Okay, we better get started again. If we can get people seated, please. I'm sorry to keep on pushing you through to one session to the other, but you will have plenty of time to interact this evening. So one of the things that we introduced this year, which wasn't there for the last 2 years, is to have plenary speakers. And our first plenary speaker today is Dr. Miles Congreve, who's Chief Scientific Officer for Isomorphic Labs. And Miles is going to tell us about using artificial intelligence to design new drugs. Now, Miles has had an illustrious career. He joined Isomorphic Labs in 2022. Previous role was CSO of Cerci Heptares, Cambridge, UK. Earlier in his career, he worked at Aztec Therapeutics. He's done very many other things, including co-inventor of a drug used in cancer. So thank you very much, Miles, and we look forward to hearing from you.
Thank you very much for the invitation, and it's great to be here this afternoon. So yeah, I'm going to ask this question. Can we use artificial intelligence to design new drugs? So we'll see if I can convince you. But first of all, let's step back and think about what we mean by artificial intelligence in this context. So really we mean deep learning, and I'll just give you a very quick tutorial. As if you're anything like me, 2 years ago I didn't know a thing about machine learning, and so there's I think quite a lot of us hear about artificial intelligence, don't really know what it is or how it works. But essentially, if we think about image recognition as a simple machine learning task, which now Google Lens will do for you and various other software, what you're looking at is an input. So here we have a car, and we're looking to categorize that input into, is it a car or is it not a car? And the way that this is done with classical ML is that you define features manually. So you say cars tend to have windows, they have headlights, they have wheels, they have a bonnet, and you extract those features in a large data set, and then you're looking for the model to learn how to classify those, and you have a training set and as well as the actual data set, and it trains on improving the validity of its output. So deep learning is a similar system, but it's one in which the feature extraction is done automatically and is broken down into many hidden layers. So it may be the pixels in the image and the colors, different shapes and angles. And the deep learning system will try to use all of that data to classify the output. And it doesn't require any manual intervention. Essentially, you set up a deep learning system, which might be a neural network, and it will then learn on the data against performance with training data. So that's what we're trying to do. So really with artificial intelligence, we need a data set and we need a problem that we can clearly define, and then we can make use of it. The other thing that I guess that's worth us remembering is that machine learning requires a lot of compute. And one of the reasons that machine learning systems are so much better than they were just a few years ago is that there is vastly more access to compute, and it's much cheaper. So you can just train models with brute force on more and more computers until it goes up another percentage and another percentage until you get a really good model. So that's something that's worth remembering. It's not all because of smart people. It's actually because of breakthroughs in access to technology from the compute perspective. So Isomorphic Labs is a spin-out of DeepMind, which is now actually part of Google DeepMind. And I'm going to back up to our legacy to give you some examples of machine learning systems and how they've been developed, and help to build confidence that we can maybe use these systems in life sciences. So originally, DeepMind looked at models for chess. Chess is relatively easy to train a model against and can be done empirically. More impressive, I think, is the example of Go. which you may be familiar with. So there's a fantastic YouTube documentary, which I'd recommend you watch, for this legendary match against Lee Sedol in 2016, where the world champion was pitted against AlphaGo, and the algorithm essentially wins that match to everyone's surprise, and 200 million people watched it live, so it's a big event. So Go, just backing up, is a 3,000-year-old game. There's about 40 million players in the East, and why it's significant is that it's too big a search environment to empirically just compute the entire space. There are actually 10 to 170 possible positions in the game of Go, which is far more than the number of atoms in the universe. In fact, there are less than 10 to the 60 atoms in the universe. So there's no way that the computer can just work out every possible move and then it knows how to do it. It has to be doing it differently. And in this legendary match, there's a famous move 37 in game 2, in which the model does something that looks completely crazy, as if it must have gone wrong. It was an innovative move that had never been really seen before. And in fact, it was that move, many moves later, that turned out to be the pivotal thing that allowed the computer to win that game. So there's evidence here that the algorithm can do things that human beings haven't done before. And I think one of the criticisms of AI is it's going to understand your data, memorize it, and just give you back the answer that you're familiar with. That's not necessarily the case. So what's it doing? So the way that the game was trained, as I implied, as a deep learning system, is that it was just set to play itself and just repeatedly, very quickly, played itself against Go, and then the win rate was the metric, the output that was looking to be optimized. And after very many iterations of this, it became very good at playing Go. So that sounds straightforward, right? So that's how it works. And what it's doing, probably, is it's navigating through the permutations, the search space that we have in Go. It can't memorize all of it, but it can say, for this move, I could make these possible choices, and which of those choices is most likely to be productive further down the game. And that's how it's learned with the embeddings, essentially, to get better and better at playing Go. And DeepMind have used this kind of sandbox of games as a safe environment to generate AI systems, with the ultimate objective of generating an artificial general intelligence model, an AGI that can be put against any problem. So it initially started with the Atari games, then AlphaGo, as I've described. AlphaZero is the next generation, where you can that one algorithm can just be given the rules of any game, and it will just play itself and learn how to play that game. That's a two-player game. And AlphaStar is then a model which can act in any kind of real-time strategy game and make decisions about how to navigate that open world scenario. So this kind of overall observation then is that we have seen, I think, the rise of foundation models. So a foundation model is where you train a model for one specific task with a certain data set, but that model is then very useful for another task. You can give it additional data and build on top of it, and it can take that broad data and carry out a general purpose. And that gets around this issue that you need to make a bespoke model for every individual process or area that you want to work on. And you can potentially just fine-tune the foundation model for the particular task that you want. I'll come back to this in a moment. So in terms of AI as a relevance to life sciences then, the next big problem that DeepMind applied its approaches to was AlphaFold, AlphaFold 1 and then AlphaFold 2. And this goes back to Christian Andersen's Nobel lecture in 1972, where he said that the three-dimensional structure of any protein should be fully determined just by its amino acid sequence. And so we have a sequence which is just like a code, and from that, that must be the physics of how that protein then folds in an ordered manner must be encoded in that code, nothing else. And that was his prediction or hypothesis. And what DeepMind then decided to do, and others, was to try and use the information available on protein folding and sequences to produce a model that could accurately predict the three-dimensional structure of a protein just from its sequence. And you can do this because there's the protein data bank, which has about 200,000 structures in it to train. And of course, you can have a test set that's not in the model and say, well, does it get this right? So you can start to build a model in a similar way to go. And of course, there's lots of ways to do this. The team at DeepMind used a lot of innovation to achieve this and a lot of compute. But in 2021, the AlphaFold 2 was published in Science and made open source to the community, and has been fantastically useful because it does generally get this right. It's not always going to get it right. It's the model. But you can take the amino acid sequence, just plug it into AlphaFold 2, and it will give you a prediction of the coordinates, three-dimensional coordinates of that protein, and you can compare it with the PDB. When it doesn't give you a fully folded structure, and you get partial folding, this may be because actually the protein doesn't fold as a single globular protein. It actually needs to be in a higher order complex. And one of the things that we've been looking at Isomorphic Labs is whether we can predict the three-dimensional structure of larger order complexes where proteins, things like transcription factors that don't give a good solution in AlphaFold 2, might actually be folded in the correct environment with other proteins inside the cell. And later on, the Protein Structure Database was published and is hosted by EMBL. So for people around the world that don't have access to compute and can't actually run AlphaFold very easily, they can actually just find their protein in the protein data bank and use that information for their basic research. You can ask the questions on the slide here. Does it look right from my experimental data? How can we use this? How can we challenge it? So this has given a lot of real-world critique of this model and allowed it to be improved. AlphaFold 2.3 includes protein-protein interactions, and you may have seen AlphaFold 3 was published recently that includes DNA and RNA protein interactions and also small molecule ligands bound to those proteins. At least the natural cofactors was made available. And AlphaFold has then been used by the community and fed back, I guess, to DeepMind in a number of other ways. So there's obviously the most logical application is structural biology. So I guess until quite recently, people would use X-ray crystallography, where you get actual crystals of the protein and you crystallize that and then you get the three-dimensional structure experimentally. More recently, last 5 years or so, there's a new technique called cryo-electron microscopy, cryo-EM, and that actually has a protein sample, and it's basically a microscope. You look at the protein in an ordered matrix, and you can get higher order complexes with cryo-EM. But cryo-EM is quite difficult, in fact, to solve the structure, and people have extensively used the predictions from AlphaFold 2 and AlphaFold 2.3 to give them the starting point to then solve the experimental data. It's been tremendously useful for the structural biology community. And then there are other examples within research projects around plastic pollution, antimicrobial resistance, etc. And what we're focused on at Isomorphic Labs is drug discovery as a particular application. So if we step back then, we know that really AlphaFold, I guess you could see as a tipping point for AI systems being relevant to some significant problem in life sciences, then what do we need to do? So I think for an AI system to be useful, hopefully from the description I've given, then we know that it can be good at finding an optimal solution from a large combinatorial space, and it can learn and make a model of that environment from the right data set. So we need the right data sets in order to generate AI systems. And we also need to have data which allows us, or a process that allows us to categorize the problem that we're trying to solve. Now, I'm a medicinal chemist, and I would say when I've done drug discovery before, it's just the creativity of my mind that has given rise to the final drug that we've got at the end of the project. And it's not a process, it's like painting, a Rembrandt or something. But in fact, it isn't. It is really a process. It's just that we don't think of it like that. We have to break down what we're actually doing step by step in the decisions that we're making, the data that we're using for each of those decisions to get to the final drug. And can we write those processes down, generate data that gives us training sets that then allows a computer to help us to do that, or potentially replace some of the steps that we need to take by using experimental data. Making compounds, testing compounds is very complex, expensive, time-consuming and slow. It takes about 5 years to develop a new potential drug and another 7 years to do the clinical research. So I think it's about asking the right questions. So I've got 3 questions here that I'm going to touch on. So the first search problem is chemical space. And this is a bit like chess or Go. It's a large space. It's thought that there are about 10 to the 60 kind of sensible small molecules that are possible, from which you should be able to select a compound that can bind to any target and give you a potential drug molecule. In fact, it's slightly more complicated than that because there are molecules of different sizes. If you want to start from a very small molecule and then build it up in steps, actually you might be able to enumerate that space. There's only about somewhere between 26 million and 1 billion compounds to think about. That's a quite small search space, so you could actually enumerate that and study that. But if you want to look at the whole drug universe, it's again too big for a fully empirical approach, and an AI system would be better to allow you to do this guided search through this landscape to get to an optimal solution. And there won't be one drug for every target. We know that for some targets there are 10 drugs on the market. There are lots of answers, lots of good solutions. So it's not quite as daunting as it sounds, because there might be 1 million different possible drugs that are in this landscape, and we just need to find one of them. The second search problem is that biology is incredibly complicated. It's much more difficult than chemistry. We understand very little about how molecules, biological molecules, interact with one another, and how they do that in a pathway or over time. Very often there's a signaling process which has a cascade. It creates a certain sequence of events, and that leads to an outcome, maybe a gene, and it is expressed. That's really difficult to model, and we're in our infancy. And one of the things that we often get asked is, well, it's not about finding small molecule drugs, it's about understanding what the targets are that we need to hit in the clinic. And that's a fair criticism, and there's a long way to go. But one of the things that we can hope for in the future is being able to describe these biological processes at more of an atomic level, where we're looking at protein-protein interactions, which we know we can approach using things like AlphaFold, and then start to understand how to improve our confidence in any given target. And it's something like one drug, for every 10 drugs that go into the clinic, only one gets to market. A lot of that failure, you could argue, is to do with competition and for commercial reasons, but quite a bit of it is because the target wasn't very good in the first place. And so relatively small improvements in our target selection could have a big impact on the clinical success rate. Even if we're only twice as good, we would get half the cost of doing drug development by improving that success rate. Things like chemical proteomics is very powerful now. This is an approach where you can generate vast data sets looking at how small molecules intervene in either with a particular protein or in a pathway, and start to map where molecules bind and the effects of perturbations on a biological pathway. So the advances of some of these very high throughput biological screening systems, coupled with AI technologies, has a lot of promise, maybe over the next 10 years or so. And I think the last problem, sort of I think there are echoes of the various panel discussions we've had this morning. There's huge amounts of data we generate in clinical studies and in clinical practice, but we're not thinking about how to use that data for AI. We're not collecting data in a consistent way. The ML scientists don't know the first thing about life sciences. They need to be given a data set from us that they can use. We can't expect them to learn how to be a clinician to then interpret the data. So we need to be able to find ways to generate data sets which then have the potential to improve diagnostics, to personalize treatment, and obviously within cancer in particular, there's already quite a lot of progress here in understanding the genetic drivers behind in individual patients' tumors, getting more information from the clinical trials themselves, having virtual placebo as part of clinical trials for assisting us in our interpretation. And I think fairly simply, things like writing regulatory submissions, and you can already get ChatGPT to give you a fair bit of content for something that you want to produce. You can, with Google Gemini, you can give it lots of PDBs and say, just use this information to generate a summary. So I would imagine that it's quite low-hanging fruit to reduce the vast amounts of time and energy we have doing the bureaucracy associated with clinical studies by using some of these AI systems. So Isomorphic Labs, ISO Labs for short, we started using AlphaFold, and we're focused at the moment on small molecule drug discovery. I'll run through this quite quickly, but this gives you some insights on how the technology is starting to impact working practices, best practice of sort of jobbing medicinal chemists like me. So the first thing is that we can now just use a protein sequence, as with AlphaFold, but now a ligand molecule description, which is actually called a SMILE string, and we can just generate one or more views or predictions of how that molecule might bind to a target or to multiple targets, etc. And AlphaFold 3 in the latest paper, and you can go to our website and find blogs and links to this, is now getting this right almost 80% of the time. So rather than having to solve an experimental X-ray crystallography or cryo-EM structure for each ligand to get that information, we can do this in silico in 5 minutes, and we can run it multiple times if we wish to get more hypotheses about how a molecule might be binding. And that vastly improves our ability to think about how we rationally design compounds. I think sort of falling out of this approach without really any specific focus, AlphaFold 3 is now quite good at protein antibody prediction, so antibody-antigen interactions. So monoclonal antibodies obviously bind to a protein target, an antigen, and this was quite a difficult problem. AlphaFold 2 only got this right about 20% of the time. AlphaFold 3 is about 52% of the time, and that's without being trained on any specific data set for protein antibody interactions. It's just from the PDB. So that would suggest that there's quite a lot of promise for these sorts of models to do de novo design for antibodies. So rather than having very large experimental screening campaigns to find an antibody, we may be able to predict a much smaller number of sequences, make and study them more carefully to make more rapid progress in antibody discovery. We're also, as I mentioned, you can look at RNA, DNA, small molecules and proteins together for things like transcription factors. So this opens up areas of target space that have been completely intractable to small molecules so far. Screening approaches, etc., haven't worked, but with a three-dimensional understanding of how the complex forms overall, we may be able to identify small molecule druggable pockets that we can target and then use algorithms to try and target those pockets. So this potentially opens up more of target space for biology to be interrogated. That's going to work. This didn't work earlier, so that's a good surprise. So this is something on our website. This is an example of a prediction for a protein called TIM3. This is from a paper from the FESIC lab. And all we're doing here is just reproducing what's known in the literature. This obviously wasn't in the training set, it's in the test set. And you could see there's quite a big movement of 2 amino acid residues in orange on binding of this molecule. And then the molecule's being grown, and each of these predictions is being made. So until, with classical methods, if you have a docking physics-based method, you wouldn't see these big protein movements, this amino acid forming a site. So this kind of induced fit or cryptic pocket that we can see where the molecule is bound now would have been very difficult to predict. But AlphaFold 3 with the small molecule added gets this right, and that's an example where obviously that paper's only just been published and it's not in the training set. So these are the sorts of things that we can now potentially do. This is another example. This is quite a difficult problem. So this is KRAS, which is obviously a cancer driver, a major target, and there's a lot of interest in inhibitors of KRAS. First drugs on the market now, and lots of additional research into inhibitors of this system. The KRAS system, initially people have targeted certain mutations that are prevalent in certain types of cancer. There's now an approach to try and get pan-RAS inhibitors that will bind wild type as well as the mutants, and that's quite difficult. So Revolution Medicine have come up with what's called a molecular glue approach, where they're finding a small molecule, which is in the center of the image, that actually encourages another protein that's generally found inside cells, cyclophilin A, as a chaperone protein, to stick to KRAS. It glues it together, and by doing that, it prevents KRAS from functioning. So these sorts of ternary complexes are very basically not represented in the PDB. So for AlphaFold 3 to understand this, work out where that interface is, correctly place the small molecule, it's also got GDP bound as a cofactor, is really quite incredible to me at least, because there's nothing in the training set to allow it to do this. It's doing this because it generally understands how proteins interact with one another. So it seems to have generalized the information that's available to it to be able to apply to completely new environments. And without going into the details, this is not a trivial problem. We have to run this 250 times. We get a distribution, and we have to score the poses. But what I can say is that the top-ranked, most confident solutions match the PDB, so the white versus the blue that came out for this system. So again, this is very encouraging for us to be able to come up with other molecular glues which might make targets that are currently intractable much more accessible, and just generally for understanding protein-protein interactions. So beyond actually predicting these structures, what we really want to do is know how well things bind, because we spend a lot of time in the lab making molecules that we hope to be more potent than the last one, until eventually you get to the most potent and drug-like molecule for your target. If we could just predict which ones were the best and put things into order, then this is essentially the holy grail of, at least from a medicinal chemist perspective, of drug discovery. And we can do this now with predictions of binding affinity at isomorphic labs. So we can now do this ranking, and essentially you're categorizing, is this molecule more or less potent than the previous one? And you can build a general model based on lots and lots of binding affinity data against many targets in order to do this. And as an example here, so this was last year actually, we took a paper that had been published by Pfizer on MC4 receptor antagonists, which is a GPCR used in cachexia. And we were able to sort this list of the active compounds from a list of inactive compounds and get 5 out of 6 of them in the top 10, essentially. So again, not perfect. This is quite a difficult system. GPCRs are very flexible with large binding sites, but the algorithm could certainly improve our confidence that certain designs would be more worth synthesizing than others and help us to filter down potential molecules that we might make to a much smaller list of things to put into practice. And then beyond that, of course, we need to know how a drug is going to be absorbed and distributed in the body. And most of the time, we have to do this by doing things like rat PK experiments or a whole suite of cell-based assays to look at various ADME predictions. So one of the things we've spent a lot of time and energy on over the last 2 years is trying to make predictions of markers or surrogates for absorption, distribution, metabolism, and excretion. We've generated a large data set to do this because a lot of the data that's available in the public domain is measured in lots of different assays with different rank orders, etc. So it's very difficult to use that data. So we've needed to generate our own data, and we can now, again, not in a perfect way, but we have statistically meaningful power within the predictions to say this molecule is much more likely to be absorbed and have good properties than this one, and that really cuts down the number of molecules you need to make in a drug discovery project. And then I mentioned that, medicinal chemists think that their creativity will ultimately design the drug. But actually, generative chemistry is quite simple. I mean, there are only so many reactions that we can carry out that are sufficiently cost-effective to be used in manufacturing. And if we're starting from, let's say, a small fragment size, small part of a molecule, and we're just building it up to fit a pocket, so it's not that difficult to automatically come up with the molecules that seem sensible and then put them through the filters that I've just mentioned. So we now have generative chemistry de novo design tools where we can generate very large numbers of molecules which are then filtered, and those molecules are chemically sensible. So when you show them to a synthetic chemist, they could, yeah, I can work out how to make that. We have retrosynthesis, which is a model which breaks the molecule down in synthetic steps and says this is going to be easy to make, this is going to be hard to make, etc. So there will be a time, I think, where you don't need to have a lot of experience of synthetic chemistry or medicinal chemistry to design drugs. You can basically use algorithms to make good suggestions of what might work, and then coupled with the predictions that we're making, we can select the most optimal ones to actually put into practice. So I briefly mentioned antibody discovery. We have, I think, quite a lot of hope that the protein-protein interaction recognition that's available to models such as AlphaFold can be used for discovery of antibodies in a much more efficient way. And we see things like ADCs in cancer as being an incredibly powerful new tool for in our armory for patients. So an example here, Generate Biomedicines, whose focus in this area, have got their first antibody into the clinic. So here we're using the sequence information, so what we know, very large databases of antibodies that do or do not bind to the target of interest, and then we can couple on top of that the structural understanding from things like AlphaFold 3, which gives some predictive power. There are other things we have to worry about, things like immunogenicity and manufacturing, etc. But again, if we generate the right data sets, and some of the pharmaceutical companies will have a lot of data in this area, we can improve our success rate, potentially make therapeutic antibody discovery something that we can be done in a few months rather than 2 to 3 years. So going back to targets just for a moment, if we think about computational biology, which is a way of using data to describe biological problems, as I said, we're in the infancy here of how we need to use or can use AI, but we can start to use data for things like genetics and other both germline and genetic changes that come in disease, epigenetics, etc. Use those data sets to give more confidence in target selection. We can start to use computational systems to look at mechanisms, to build support, to say, well, we've got all this data on this individual protein. What about the actual pathway that it sits in? Is there evidence for another protein in the same pathway? Does that build our confidence that this is a good mechanism? There's already a drug upstream. Will this therefore be a useful drug? In particularly important is predicting toxicity and selectivity. The selectivity of a compound you can see is just another measurement of the binding affinity of that molecule to a protein. But instead of just looking at your protein of interest, you're perhaps looking at the whole genome or a few thousand genes that we know are associated with issues for tolerability and toxicity. So it may be ion channels and GPCRs in the heart, for example, would be good targets to make sure you're not binding to, and can use that as a filter. And you can also use models of cell-based cytotoxicity, start to try and generate models to say, this molecule is likely to be cytotoxic, we shouldn't make it. This one is very unlikely to be cytotoxic. And then as we move into the clinic, I'll come on to this in a moment, we can start to use these sort of computational biology approaches to drive how we do preclinical validation, try to develop systems preclinically that are very similar to the ones we're going to measure in the clinic and look for that direct translation. And I think critically importantly, trying to come up with biomarkers, things that we can measure very early, potentially in volunteers in the clinic. So another DeepMind model is AlphaMissense that was published last year. This is a model that predicts whether the missense mutations that we all have in our genome are likely to be pathogenic or not. So if you then had your genome sequence, the missense model would say, of course you've got lots of variations in your particular genome, there may be some missense mutations that would give you a higher chance of having disease in the future. And so this is one of the data sources that you can use in target selection, for example, to say there seems to be an association between missense for this target, for this disease, is that therefore a drug target? So this is one of the inputs that you could use. So just to wrap up then, going back to the clinic, I think, again, this comes down to us thinking about the data we're collecting and the way that we do it, and how we're not considering each clinical trial in isolation, but how we're doing things across different studies. And we may be able to use the data that we're generating much more intelligently to help us with safety signal detection, modeling of previous trials, and which ones were successful and which ones were not. Obviously, patient selection, if you've got any type of markers or missense or genetics, is a very powerful way of getting a better outcome. Trying to predict model efficacy, actually you can, in silico, try and look at the outcome of different clinical trial designs to see which is most likely to be successful. So there's a lot that we can do here, and I think it's really a communication between the people generating the data scientists and the ML scientists, and that's where the human creativity is required. And the FDA last year published quite a detailed article on this, so they obviously recognize this as an opportunity and have thoughts on what they would like to see. I think then finally, in manufacturing for the small molecule drug itself, often it can be very cost ineffective and very slow to generate the API that we actually need to do a clinical trial. It contributes to this valley of death that we have in smaller companies that find it difficult to fund getting clinical studies done. If we can come up with much better routes and potentially create new reactions, actually use AI to optimize the chemistry itself, then we can come up with shorter, more cost-effective, more robust routes with better control of impurities and a better specification. So there's a lot of potential here, and actually chemistry, as I said, is much simpler than biology. It's much easier to generate data on a particular reaction. You can generate tens of thousands of permutations of that exact reaction and use that to generate a model to optimize that particular reaction or similar reactions. So this should be quite tractable. And again, we measure vast amounts of information in the synthesis and drug manufacturing process, and if we can use that information more intelligently, then we can actually improve many of the kind of quite boring things that we have to worry about in regulatory compliance and things like shelf life and supply chain. So it might be quite dull, but because these areas where there's tons of data, actually it should be relatively easy to make improvements. All right, so I'm going to stop in a moment. I think I may or may not have convinced you today, but I think AI is coming for drug discovery, whether we like it or not, in some way. And if it's not today, it will be in 10 years' time. And I'll close there, take any questions.
Thank you very much. Miles has agreed to take a few questions, so please. Can we have a microphone, please?
Chris Finnis from Phenotypica. How do you see the scope of AI perhaps repurposing some already approved drugs in combinations to get some quick wins?
Yeah, that's a great question, and I've got a longer lecture with a section on that. Yeah, I think that fundamentally I would say that drug repurposing is quite a challenging thing to do because the drug's been developed for a very specific purpose, and the idea that it's just going to be perfect for another indication is a bit of a leap of faith. But on the other hand, there may be repositioning of drugs into a more specific patient population, which would have a better chance of success. I think that's very likely to be possible. And drug combinations, particularly in oncology, I think is an area where we could probably do much better by modeling things preclinically and using AI systems to help us predict the chance of success of a particular combination or particular dose frequency or whether you dose together or sequentially and things like that. You could potentially model that. I think there's a lot of potential for that. It's not something that we're doing, but I think it's an area that I think with the right data sets, you can make a lot of progress.
Okay, I can see several more hands up, so please, you next.
Thank you for the talk. Ron Jortner, Aspire Biosciences. With early neural networks that just had a handful of layers, let's say, you could basically look at the network after it converged onto a solution and understand the mechanism of the solution. Obviously, with deep learning, with several thousands of networks, very often you reach a really good solution, but you struggle to understand how it works. Obviously, with drug regulation, it's been going the other way. So nowadays, drugs that come off phenotypic screening without a mechanism are not going to be approved. So how do you bridge that gap? How are you going to deal with the fact that you're going to get solutions that work beautifully but might be difficult to explain?
Yeah, I agree that would be challenging if you were using it to just optimize a drug without understanding how that drug or drug combination is working. But many of the things that I've been talking about are processes on the journey to find a drug in the first place. You know, find a drug against a known target, but one where small molecules are very difficult to develop, or just to run projects much more quickly and efficiently. So in that case, once you generate the model, you're actually immediately then demonstrating that prediction is correct in a specific context. So that would not be an issue then from a regulatory perspective, even if we don't understand how the model is making the predictions. I think it's correct that if you have a model that's more holistically making a prediction and you don't understand how it's suggested a particular molecule for a particular disease, you'd then have to go away and unpick that, try and work out what that molecule's doing, maybe by using proteomics, for example, to then go back and say, well, it's binding to this combination of targets.
You said that you felt you had to go and generate your own data set in the ADME area. So presumably you went through a systematic view of the data sets, saying these ones are fit for purpose, these ones aren't. How did you do that? And where were those areas that you either wish there was a better data set or felt almost the need to generate your own?
Yeah, so what we recognize, I think, is that there's a lot of data, but for any given molecule, there tends to be one or two cell-based assays, for example, whilst what we actually need is to have a complete description of each molecule against a full range of parameters, because these different properties interrelate. So if we just know the solubility and lipophilicity of a compound but nothing else, and another compound we know its permeability and clearance, then that doesn't teach us very much. But if you have, what we've done is generated a large data set where we've measured all the parameters against every compound. So that gives a much richer description of each molecule, which we think will be more useful for training. And that's been very expensive and quite time consuming. But if we're seeing evidence that it works, and one of the things that you can do with these models, you can say, well, it's produced this degree of benefit to the model. So if we spend another $50 million, then we could get the solution, right? So it allows you to work out what it would take to scale to get to the end. Although that's assuming it's linear, it's not always linear.
Okay, just last two questions to yourself and over there.
Okay, thanks, Miles. That was really great talk. Just so, not surprisingly, an antibody question. So you had success rates of, I think, 20 going to 51% for predictions for antibody binding. Is that based on a known antigen and the paratope binding, or is that completely agnostic? And what are your sort of kind of confidence levels that we might be able to get to agnostic, without knowing originally what antigen the antibody was targeted to, whether we could predict that?
Yeah, so the problem with what we're looking at is you have a given antigen and a given protein, and how is that complex formed? And the measurement is whether it's within 2 angstroms RMSD of the interaction. So if it's binding clearly on somewhere completely different, then it would fail. If it's significantly out in terms of the interaction, then it would fail. So it's very much focusing just on that protein-protein interaction. But because we can rank things and give confidence, then you could ask that question. You could say, well, here's a ton of antigens and here's a ton of antibodies, which ones bind to which partner, and give a ranking and a confidence in that. So that's something that we're starting to look at, and a lot of people like Generate have been working on for some time.
Okay, last question.
Sure. I was just curious about the underlying assays often have their own flaws, but obviously you have data from these assays. But when you take more of a multi-parameter approach, are you able to kind of have more signal to noise that might come from any single assay that may have its own flaws inherently, if that makes sense? Like, do the assays themselves impact the quality of the data, and how do you actually account for that?
Yeah, I mean, that's a great question. I mean, there's one of the biggest problems in life sciences that the data is very noisy. But what you can do is, if you're using categorization as an approach, it depends what your question is. So as a medicinal chemist, do I care exactly what the protein binding of drug is? Not really. I just want to make sure it's not 99.999. It just needs to be a measurable figure that allows for some free drug exposure. So the question is not exactly what's the protein binding. It's like, is it really awful or is it okay? And that's like, is it a car or is it not a car? So although the data quality may be lower, it may be sufficiently good to allow a model to categorize. And then if you imagine a big set of traffic lights, then you can say, well, I'm looking for greens for everything. And it doesn't really matter whether if it thinks it's 95% and it's really 40%, because that's not really relevant to what we're trying to do. So that's partly how we get around the problem of low quality data. And there are other denoising effects like diffusion type models as well that you can use that take account of noise.
Okay, well, thank you very much, Miles. I'm sure there are many more questions. I have a few, but maybe I can catch you in coffee to ask those. But thank you very much. Fantastic talk.